Hello guys, welcome. Welcome to our Ashutosh coaching center. Now today we are going to start chapter number 11 and the name of chapter is biotechnology and the principles and processes. Okay, now starts. Biotechnology deals with the technique of using live organism or enzymes from the organism to produce product and of the processes useful to humans. In this case, making curds, breads, or wine too, and which are all microbes mediate processes could also be taught as a form of biotechnology. However, it is used to restricted sense today to refer to such of those processes which use genetically modified organism to achieve the same of on a larger scale. Further, many other processes techniques are also include under the biotechnology. For example, in vitro fertilization leading to test tube baby and synthesizing a gene and using it, developing DNA, developing a DNA vaccine or correcting or detect defective gene are all part of biotechnology. The European Federation of Biotechnology (EFD) has given definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follow: the integration of natural science and organism cell parts therefore thereof of and molecules, the analog for products and services. 11.1 Principle of biotechnology. Among many and two core techniques that enable birth of modern biotechnology are the genetic engineering technique to alter the chemistry of the genetic material DNA and RNA. Now here to introduce these into the host organism and thus changes the phenotypic of the host organism. Bioprocess engineering, the maintenance of sterile microbial contamination free ambience in the chemical engineering processes to enable growth of any desire of only the desired microbes eukaryotic cell in the large quantity of manufacture of biotechnological product like antibiotic and vaccine and enzymes etc. Let us now understand the conceptual development of the principle of genetic engineering, and you probably appreciate the advantage of sexual reproduction over the sexual reproduction. The former provides opportunity for the variation for the formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. A sexual reproduction preserves the genetic information, while the sexual reproduction permits variation. Traditional hybridization procedure used in the plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired gene. The technique of genetic engineering, which include creation of recombinant DNA and the uses of gene cloning and gene transfer, the overcome of this limitation and allows us to isolate and introduce only one or a set of desirable genes without introducing an undesirable genes into a target organism. Do you know the likely the fate of a piece DNA which is somehow transferred into a alien organism, mostly like the this piece of DNA would not able to multiply itself in the progeny cell of the organism, but when we get integrated into genomes of the recipient recipient, it may multiply and hereditary inherited along the host DNA. This is because the alien species of DNA has become a part of a chromosome, which has a ability to replicate. And in chromosome, there is a specific DNA sequence is called the origin of replication, and which is responsible for the initiating replication. Therefore, for the multiplication of any alien piece of DNA in an organism, in it needs to be a part of a chromosome, which has a specific sequence known as the origin of replication. Thus, an alien DNA is linked with the origin of replication, so that this alien piece DNA can replicate and multiply itself in the organism. And this can also be called as cloning, or making multiple indicate copy of any template DNA. Let us focus now. Let us focus on the first instant of the construction of an artificial recombinant DNA molecules. The construction of the first recombination DNA emerged from a response from the possibility of linkage a gene encoding antibiotic resistance with a native plasmid, autonomously replicating circular extrachromosomal DNA of Salmonella and the Trifimurium. The, the Stanley Cohen and Herbert Boyer accomplished this in 1972 by the isolating the antibiotic resistance gene by the cutting out a piece of DNA from a plastid, which was responsible for the concerning antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of the so-called molecular scissors restriction enzymes. Restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. The plasmid DNA act as a vector to transfer of piece of DNA attached. You probably know the mosquito act as an insect vector to transfer the malaria parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which act on cut DNA molecules and join their ends. This makes a new combination of circular or autonomously replicating DNA create a in vitro and is known as recombination DNA. When DNA is transferred into Escherichia coli and a bacterium closely related to the Salmonella, it could replicate using the new host's DNA and the polymerase enzymes and make multiply copies, and the ability to multiply copies antibiotic resistant gene in the E. coli was called cloning of antibiotic resistance. And in E. coli, you can hence infer that there are the three basic steps in genetically modifying an organism: identification of DNA with desirable gene, introduction of the identified gene into the host, the maintenance of the introduced DNA in the host, and the transfer of the host of its progeny. Now, 11.2 tools of recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering recombinant DNA technology can be accomplished only if we have the key tool, that is restriction enzymes, polymerase enzymes, and ligase vectors and the host organism. Let us try to understand some of this detail. 11.2.1 restriction enzymes. In this year 1963, the two enzymes responsible for the restricting the growth of the bacteriophage in Escherichia coli were isolated. One of these added methyl group of DNA, good DNA, and while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease and the Hindi second, whose functioning depend on a specific DNA nucleotide sequence was isolated and the characterized 5 years later. It was found that Hindi second always cut DNA and the molecules at a particular point by the recognizing a specific sequence of six basic base pair. This specific base sequence is known as the recognition sequence for Hindi second. Beside Hindi second, today we know more than 900 restriction enzymes that have isolated for over 230 strains of bacteria, each of which recognize different recognition sequence. The convention for the naming these enzymes is the first letter of the name comes from the genus and the second two letters from the species from the prokaryotic cell from which they were isolated. Example: E. coli comes out from Escherichia coli ry, and 13 point in E. coli the letter R is derived from the name of letter R is derived from strain, and Roman numbers following the names indicate in the order in which the enzymes were isolated from that strain of bacteria. Restriction enzymes belongs to a larger class of enzymes called nucleus. These are two kinds of nucleus: exonucleus, endonucleus. Exonucleus remove nucleotide from the end of the DNA, whereas endonucleus may cut at a specific position within DNA. Each restriction endonuclease function by the inspecting length at a DNA sequence. Once it find its specific recognition and sequence will bind to the DNA and cut each of the strands of the double helix at a specific point in their sugar phosphate backbones. Okay, now figure 11.1, each restriction into exonucleus and recognize a specific palindromic nucleotide sequence in the DNA. External restriction enzymes, these are vector DNA, and these are also important for the point of view of examination. You should note this, okay? And I should also screenshot. I should also take a screenshot of this image, okay? Now this is the a step in the formation of recombinant DNA by the action of restriction in the exonuclease enzymes E. coli. Okay, now, no. Do you know that? Do you know what the palindromes are? And these are a group of letters that forms of the same words when read both forward and backward. Example: Malayalam. That's a great example. Great man. मलयालम इज द नेम, जिसको आगे से पढ़ो या पीछे से पढ़ो आता सेम ही है. As against the word palindrome were the same word in both direction. The palindrome DNA is sequence of base pair that read same on the two strands when the orientation reading is kept the same. For example, the following sequence read the same on the two stand 5 through 3 direction. This is also true if read 3 to 5, like a 3 to 5 to 3 is equal to Gatic and 3 to 5 is equal to Kedec. This is just a opposite word. A restriction enzymes cut the strands of DNA little away from the center of petrosome. Yes, petrosome sites, but between the same two bases of on the opposite strands, this these leaves single stranded portion at the end. There are overhanging stretch called sticky end on the each strands. Figure 11.1. These are the named so because they form hydrobond with their complementary cut counterparts. This thickness of the end facilitates the action of enzymes DNA ligase and restriction of endonuclease are used in the genetic engineering to form recombinant molecules of DNA which are composed of DNA from the different sources genome. When cut by the same restriction enzymes, the resultant DNA fragments and have the same kind of sticky end and these can be joined together end to end and using ligase. These are also important. Okay, this is the important for the examination. You should note this. Okay, this is very sorry. This is the very important. हाँ, इसका स्क्रीनशॉट भी हो गया है। अब आगे देख लेते हैं हम लोग। इसे इसका आप भी लोग स्क्रीनशॉट ले ले, बेटर होगा। ओके, इस सब का आप स्क्रीनशॉट ले ले तो ज़्यादा बेटर होगा, बिकॉज इस एग्ज़ाम में तो आ ही सकता है मे बी। ओके, और नीड कुछ गारंटी होता नहीं है। चलिए आगे देख लेते हैं। आप पढ़ते रहिए और जहाँ जितना दिखे उतना ज़्यादा उसको याद करने का कोशिश कीजिए। ये जो आपको दिख रहा है इसका भी मे भी हो सके तो एक छोटा सा पिक बनता है, ना हो सके तो आप इसका वॉलपेपर ही लगा लीजिए, ये बहुत काफ़ी इंटरेस्टिंग है। ओके, हाँ हाँ, नाउ now starts. You may have realized that the normally unless one cuts the vector and the source of DNA with the same restriction enzyme, recombinant vector molecules cannot created and the separation and isolation of DNA fragment. The cutting of DNA by the restriction endonuclease result in the fragment of DNA. These fragments can be separated by the technique is known as the gel electrophoresis. Since DNA fragments are negatively charged by molecules, they can be separated by the forcing them to move towards anode under an electronic field through the medium matrix. Nowadays the most commonly used to matrix agarose, which is a natural polymer extracted from a seed. The DNA fragments separate resolve according to their size through the seizing effect provided by the agarose and gel. Hence the smaller fragment size, the farther it moves. Look at the figure 11.3 and the gauge at which end the gel sample was loaded. This was the okay. Figure 11.3, a typical aerosol gel electrophoresis showing migration of undigestive lanes and uh, digestive set of DNA fragments lanes 2 to 4. Okay, now starts. The separate DNA can be visualized only after staining of DNA with a compound known as the ethidium bromide followed by the exposure to UV radiation. You cannot see pure DNA fragment in the visible of light and without staining. You can see bright orange colored band of DNA in a ethidium bromide staining gel exposed to the UV light. And figure 11.3, the separated brand of DNA are cut off from the aerosol gel and extracted from the gel piece. This step is known as the elution, and DNA fragment purified in this way and are used in the constructing recombinant DNA by the joining them with cloning vector. Okay, now what is cloning vector? Cloning vector देखा जाए होता क्या है, सबसे पहले देखेंगे. You know that the plasmic and the bacteriophage have already replicate within the bacteria cell independent of control of chromosomal DNA. Bacteriophage, because of their high number per cell, have very high copy number of genomes within bacteria cell. Some plasmid may have only one or two type 2 copies per cell, whereas other may have 15 to 100 copies per cell. The numbers can go even higher if we are able to link an alien piece of DNA with bacteriophage or plasmid DNA. We can multiply its number and equal to copy number of plasmid or bacteriophage. The vectors used at present the engineered in such a way that they help easy linking of foreign DNA and the selection of recombinants from the non-recombinants. हाँ, दिस इस फिगर को आप बड़े आराम से नोट कर लीजिए, काफ़ी इंपॉर्टेंट फिगर है. ओके, दिस इज द फिगर. ओके, एंड आई थिंक इसका फिगर का फोटो तो बनता है एक. भाई इसे बिल्कुल सेल्फी की तरह मानो इसको. ओके, बहुत सारा डायग्राम आएगा बट डजंट मैटर. Now starts. The following are the features that are the required to facilitate cloning into a vector. Origin of replication, ORI. Origin of replication को short form में ORI कहते हैं. This is a sequence from where replication starts, and any piece of DNA when linked to the sequence can be made to replicate within the host cell. The sequence is also responsible for the controlling the number of the copy number of the linked DNA. So the if one want to recover many copies of the target DNA should be cloned in a vector whose origin support high copy number. Selectable marker word in addition to ORI, vector requires a selectable markers which helps to identifying the eliminating non-transformants and selectively permit the growth of the transformant. The transformation is procedure of through which a piece of DNA is introduced in a host bacterium. You will be study process in the subsequent selection. Normally, the gene encoding resistance to antibiotics such as ampicillin and chloramphenicol and the tetracycline or the chemacycline etc. are the considered useful to the selectable markers for E. coli, and the normal E. coli do not carry resistance against any of these antibiotics. Second, cloning site. In order to link the aligned DNA and the vector need to have a very few preferable single anti-conjugation site for the commonly used restriction enzymes. Presence of more than one reconjugation sites within the vector will generate several fragments which will complicate the gene cloning. Figure 1.11.4. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. For the example, you can ligate a foreign DNA at a Bahama site of tetracycline resistant gene in the vector PBR 322. बूम द बूम इसको नोट कर लीजिए. पी वेक्टर 322. काफ़ी बाई चांस लॉक हो गया तो डरने की बात तो नहीं है. हाँ, तो ये हमने ले लिया है पहले से स्क्रीनशॉट और कोई बात नहीं है. The recombinant plasmid will lose tetracycline resistance due to the insertion of the foreign DNA but can be still be selected out from the non-recombinants one by plating the transformants on tetracycline containing medium and the transformation growing on a ampicillin containing medium or the transform on a medium containing tetracycline recombinants will be grow in the ampicillin containing medium but not on the containing tetracycline, but non-recombinants will grow on the medium containing both antibiotic. In this case, one antibiotic resistance genes help in the selecting the transformants, and whereas the other antibiotic resistance genes get and inactivated due to the insertion of aligned DNA. and help in the selection of recombinants. Selection of the recombinants due to the inactive antibiotic is a cu- cumbersome and the procedure because if required simultaneously plating of two plates having differently antibiotic. Therefore, alternative selectable markers have been developed from the differentiate recombinants from the non-recombinants on the basis of ability to produce color in the presence of chromosomatic substrate. In this, a, a recombinant DNA is inserted within the coding sequence of enzymes G galactosidase, and this result into a inactivation of gene for the synthesis of these enzymes, which is referred to the insertional inactivation. The presence of chromosomatic substrate give us blue colored colonies of the plasmid in the bacteria does not have an insert. The presence of insert result into the insertional inactivation of the B galactosidase gene, and the colonies do not produce any color. These are these are the identified recombinants and colonies vector for the cloning gene in the plant and animal. You may surprised to know that we have learned the lesson of transforming genes into the plants and animals from bacteria and virus, and which have known these four legs, how to deliver genes to transform eukaryotic cells and for them to what bacteria or viruses want. For example, ang- agrobacterium agrobacterium and हाँ इसको थोड़ा बड़ा कर लेते हैं. हाँ अब बढ़िया से दिखेगा. Agrobacterium and tumefacian, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA and to transform a normal plant cell into a tumor and direct these tumor cell produce chemical required by the pathogen. Similarly, retrovirus in animals have ability to transform normal cell into a cancerous cell. Okay, this is a borderline retrovirus wala. Okay, okay. Better, a better understanding of the part of the art of delivering genes by the pathogens in their eukaryotic host has generated knowledge to transform these tools of the pathogens into a useful vector for the delivering genes of the interest of you to human. The tumor including TI, the plasmid of Agrobacterium tumefaciens, and has been modified into a cloning vector which is not more pathogenic to the plant but still able to use the mechanism deliver genes of our interest into a variety of plants. Similarly, retrovirus have also been disarmed are the used to deliver desirable genes into animal cells. So once a gene or DNA fragment have ligated into a suitable vector, it is transferred into a bacterial plant or animal host where multiplies. 11.2.3 competent host means to say that formation for the transportation transformation with the recombinant DNA. Since DNA is a hydrophile molecules, if cannot pass through cell membrane wide in order for to bacterium to take up the plasmid, the bacterium cell must be made competent to take up DNA. This done by treating them with a specific concentration of a divalent cation such as calcium, which increases the efficiency with which DNA enters. Now, the bacterium through pores in its cell wall recombinant DNA can then be forced into a such cells by the incubating the cell with the recombinant DNA on ice, followed by the placing them briefly at 42 degrees Celsius heat shock and then putting them back on rice. They enable that bacteria to take up the recombinant DNA. This is not only way to introduce LNDNA into the host cell in a method known as microinjection. Recombinant DNA is directly injected into a nucleus of an animal cell. If in other methods suitable for plant cells are bombarded with high velocity microparticles of gold tungsten coated with the DNA method is called biolistic gene. This is very important for the point of view of the examination because it's a completely unique question. And the last method is used to disarmed pathogens vector, which when allowed to the infect the cell transfer the recombinant DNA into the host. Now that we have learned about the tool for the construction recombinant DNA, let us discuss the process facilitating recombinant DNA technology. 11.3 The processes of the recombinant DNA technology. Recombinant DNA technology involves the several steps in specific sequence such as isolation of DNA and the fragmentation of DNA by the restriction endonuclease, isolation of a desired DNA fragment, ligation of a DNA fragment into a vector, transforming recombinant DNA into the host, and the cul- culturing the host in a medium at a large scale, extraction of the desired product. Let us examine each of these steps and in some details. Okay, this is a DNA separation, remove spooling. कुछ इस तरह से स्पूल को रिमूव किया जाता है. Look at this. Now let's take isolation of genetic material DNA. Recall that nucleus is the genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. On order to cut the DNA with restriction enzymes, it is need to be free from the other macromolecules since the DNA is enclosed within the membrane. We have to break the cell open released to DNA and along with the macromolecules such as RNA, proteins, and the polysaccharide and also lipid. This can be achieved treating by the bacteria cell and the plant cell or animal tissue with the enzymes such as the lysozyme, bacteria, cellulose, and plant cell, and chitinase is in fungus. You know that the genes are located on the molecules of DNA intertwined with the proteins such as histones and RNA can be removed treatment and with the ribonucleic acid nucleus, sorry, various proteins can be removed and treatment with the protease and other molecules can be removed by the appropriate, okay, yes, appropriate treatment, and the purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as the collection of fine thread in the suspension 11.5 figure. This is the figure, okay. इसका स्क्रीनशॉट आप ले ले तो ज़्यादा बेटर होगा. This and now इसका भी पहले ही स्क्रीनशॉट ले ले तो ज़्यादा बेटर होगा. हाँ, this is the okay. That's a great. Now रात के 11:42 हो गए हैं तो आगे देख लेते हैं हम क्या है 11.3.2. ज़्यादा कुछ नहीं, आप लोग लाइक और सब्सक्राइब कर दें जिससे काफ़ी हम लोग को सपोर्ट मिलेगा और कुछ तो होगा नहीं. हाँ देखते हैं 11.3.2 द कटिंग ऑफ डीएनए एट a specific location. Restriction enzymes and digestion are performed by the incubating purified DNA and molecules with the restriction enzyme at the optional condition for that specific enzyme ag- agarose and gel and electrophoresis and is employed to check the projection of a restriction enzyme's digestion. DNA is negatively charged molecules, hence it moves towards the positive electrode anode and the 11.3 the process repeated with the vector DNA is also the joining of DNA revolves involves the several process after cutting the source DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of the interest from the source DNA and the cut vector with the space mixed and the ligase is added. The result is in the preparation of recombinant DNA. 11.33 amplification of gene of interest with using PCR. PCR stand for the polymerase chitin reaction, and in this is the full form. In reaction, multiply copies of the gene or DNA interested synthesized in vitro too. Okay. Hmm. A set of primers, small chemically synthesized all oligonucleotides that are complementary to region of DNA and the enzymes DNA polymerase. The enzyme extend the primers using nucleotide and provides in the reaction and the genomic DNA as template. If the process is replication of DNA repeated many lines and the segment of DNA can be amplified approximately billions times, that is 1 billion copies are made, and such as repeated the amplification is achieved by the uses of thermostable DNA polymerase isolated from the bacterium thermus and the aquaticus, and which is remains active during the high temperature induced denaturation, okay, of double standard DNA. The amplified fragment, if desired, can now be used to ligate with the vector for further cloning. Figure 11.6. 11.3.4 insertion of recombinant DNA into the host cell organism. There are several methods for introducing the ligated DNA into a recipient cell and the recipient cell after making them competent to receive take up DNA present in its surrounding. So if recombination DNA bearing gene for resistance to antibiotic, example ampicillin, is transferred into E. coli cell. The host cell become transferred into penicillin resistance. If we spread the transferred the cell on agar plates containing penicillin, only a transformants will grow and the untransformed recipients cell will die, since due to the ampicillin resistance gene one is able to select transformed cell in the presence of ampicillin. The ampicillin resistance gene in this case is called selectable marker. 11.3.5 obtaining the foreign gene product. When you insert piece of aligned DNA into a clotting vector transfer into a vector the plant or animal cell, the aligned DNA gets multiplied. Almost all recombinant techniques ultimate aim to produce a desirable protein. Hence, then there is a need for the recombinant DNA to be expressed. The foreign DNA gene gets expressed under appropriate condition. The expression of foreign genes in the host evolved understanding many technical detail. After having cloned the gene of interest, having optimized condition to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think any reason why there is need for the large scale of production? If any protein encoding gene is expressed in a heterologous host, is called recombinant protein. The cell harboring cloned and the genes of interest may grow on a small scale in the laboratory. The culture may be used for extracting desired protein and the purifying it by the using different separation technique. The cells can also be multiplied in a continuous culture system wherein used medium is drained out one side while fresh medium is added from the other, maintain the cell in their physiological most. Hmm. The active log exponential base phase, and this type of culturing method procedure produces a larger biomass leading to a higher yield desired protein. A small volume culture cannot yield appreciate quantity of product to produce in large quantity. The development of bioreactor where uh, large volumes 100 to 1000 liter or culture can be processed was required. Thus bioreactors can be pouched of a vessel in which in which raw are biological converted into a specific product. Individual enzymes etc using microbes, plants, animals or human cells. A bioreactor provides optimal condition for achieving the desired product by the providing optimum growth condition, temperature, pH, substrate and salt, vitamins, oxygen. The most commonly used bioreactor are stirring type which are shown in figure. These are the figure which is very important on the point view of for the नीट. ये भी नीट के पॉइंट ऑफ व्यू से बहुत इंपॉर्टेंट है. Now it's done. No, it's not done. Now it's done. Okay. चलो आगे देखते हैं. हाँ. A stir tank reactor is usually cylindrical or with a curved base to the facilitate the mixing of reactor content. Stir the facilitate even a mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has alligator system, an organism deliver system and a form control and a temperature control system, pH control system and the sampling port so that the small volume of the culture can be withdrawn periodically. 11.3.6 Downstream processing. After the completion of the biosynthetic stage, the product has to be subjected through a series of process of before it is ready to marketing as finished products. The processes include the separation and the purification have are collectively refers to as downstream processes, and the product has to be formulated with suitable preservatives and such formulation has to be undergo through in clinical trials and in drugs as in case of drugs. Strict quality controlling the testing for the product is also required. The downstream processing and the quality control testing vary from the product. Summary. Biotechnology deals with a large scale and the product marketing of product and processes on the living organism and cell enzymes. And modern biotechnology using a genetically modified organism was made possible by the man learned after alter chemistry of DNA and conduct recombinant DNA, and this key process is called recombinant DNA technology or genetic engineering. Okay, and the process involves the uses of restriction endonuclease and DNA ligase and associate plasmid or vital vectors to isolate and ferry the foreign DNA into the host organism, expression of the foreign gene and the purification of gene product that is functional protein, and the finally making a suitable formulation for the marketing and the large scale production involves use of bioreactor. So guys, thanks you for the watching video till the end, and you are a part of NEET, so please like and subscribe and share mostly, okay, as you can do, जितना हो सके इसे उतना ज़्यादा शेयर करें. बाय बाय, सी यू इन नेक्स्ट वीडियो, बाय.